Very good evening and a warm welcome to all our viewers. I'm Dr. Shatil from the Clinical Engagement team here at DocPlexus. Welcome to our World Malaria Day special webinar. Globally, 3.3 billion people in 106 countries are at risk of malaria. In 2012, malaria caused an estimated 627,000 deaths, mostly amongst African children. It gives me great pleasure to welcome you all to this important gathering as we mark World Malaria Day 2025, a day of reflection, action, and renewed commitment in the global fight against one of the oldest and deadliest disease known to humankind. This year, the theme is both a call to action and a powerful reminder of our collective responsibility. Malaria ends with us. Reinvest, reimagine, and reignite. We have an expert with us who will share insights on malaria control, emerging research, and strategies to drive us closer to elimination. Dr. Amitabh Banerjee is the Professor Emeritus Epidemiology at Dr. D.Y. Patil Medical College, Pune, India. Dr. Amitabh Banerjee served in the Indian Armed Forces from 1978 to 2005. He started working as a professor and subsequently as the head of Department of Community Medicine at Dr. D.Y. Patil Medical College, Pune. Dr. Banerjee has published over 100 papers in national and international journals and has undertaken several funded research projects. He has also delivered several keynote addresses at national and international conferences. It is an absolute honor to have him here with us today. Without further ado, let's welcome Dr. Amitabh Banerjee.
Good afternoon and greetings from Pune. So today we'll be talking about the malaria control, basically vector control and vaccines, and what is the future of malaria prevention, and whether are we in the end game, that is whether we will be able to eradicate malaria as we once thought long back. So I'll be trying to give the context because this recent advances in vector control and the vaccines, these are only part of the solution. Malaria is a very complex disease, and I'll try to recapture some of it, giving the history of malaria and its attempt at eradication. And so that way I will be not able to change the sound. Yeah, I am. So historically, I'll be going back. It is not been amenable to eradication, and we have failed, and we'll see the reasons so that we do not make the same mistakes again. And we will see the social factors which contributed to failure of eradication. And again, I think we are going too much skewed towards technological solutions, which we did in the past. And again, if we don't take into consideration the social factors, these impact of technology like new vector control measures and new vaccines will just graze the surface of the problem. And tribal malaria is a major issue in the eradication of malaria. Any foci, any focus which remains, and most of these foci remain in the tribal belts because they have got their own ecology, they have got their own vectors, they have got their own dynamics. And it can, malaria can make a resurgence if any of them is left unattended. And because of the complexity of the problem, it is not easy. So we'll come to that. And it is not only the hard sciences, like you have got a parasite, you have got anti-malarials, then you have got vectors, different types of mosquitoes. There are many types of different mosquitoes, and they vary from region to region. Even in the same country, the vectors are different. So all this make the thing very complex. So that each one different vector have got the different resting habits, which has a bearing on the say insecticide spray or say whether they will be amenable to control. So we have to lot of terrain, lot of geographical differences. So all these come, and similarly, a lot of human behavior. Human behavior also vary. The tribals, they have their own way of living. All those affect, and then we have got, besides the tribals, the lot of population movements in a vast country like India. And uh, another thing which has been missed out in the past, which we have occasion to study, has been the exposure to the elements. And one of the major area where there is very little mention is the open air toilets in the forest or toilets in the wilderness, which are um, exposes the humans to mosquito bites. So this we have to see the sociological factors, and even a small risk in a, say, where open air exposure to large number of people will maybe a small risk, but if the whole population is exposed, the risk becomes high, the incidence becomes high at the population level. So in clinical medicine, William Osler said that one who knows syphilis knows medicine. So I have a feeling that in public health, in epidemiology or preventive medicine or social medicine, in public health, one who knows malaria knows public health, because the problem of malaria is not straightforward. There are a lot of regional variations, lot of terrain variations, and we'll see. I'll just give most of the my experiences had been in the armed forces, and being in the armed forces, I have had occasion to work in tribal areas, because our troops are deployed in interiors, in forest, in the northeast and in central India. I have had occasion to see malaria dynamics firsthand. And so, I'll, even from a country like India, we have a lot of experience which can just give you some example of the complexity of the problem. So, I have a tendency to compare the malaria epidemiology with the game of chess. So, because like in chess, they have got different pieces, and the combination of pieces wins or loses a game. And if you see in the chess left hand side, I have marked the four center squares. That means if anybody who has a preliminary idea of chess will know, the one who controls the center of the board and the mobility of the pieces, and the all the pieces are working in harmony, that person can only win the game of chess. If that person plays with one new piece like a queen or a EM and neglects the other pieces, so amateurs, they play with the big pieces. So similarly, any new discovery in malaria control, whether it is insecticide, whether it is bed nets, or whether it is now vector control measures, genetic interventions, or newer vaccines, the there is a excitement and like an amateur chess player, and all focus is on that one piece only, the new piece only, and neglecting the other pieces. And because of the complexity, perhaps malaria is more complex than chess also. As I said, each vector has got its own habits, own resting places, just like each piece of chess. Knight moves in a different way, the bishop moves a different way. So anyway, this is just to give you a rough idea of the complexity of here. And one has to do a lot of field work and study the behavior of humans, study the behavior of mosquitoes, uh, to plan a control measure. So this I gave you a picture of a chessboard. If you see the picture of India, these green areas are the highly malarious areas. These are forested areas, tribal areas. So similarly, if you see in the center India, there is in the center, they are the green areas. That is just like a chessboard. Once who controls the tribal areas, malaria. And similarly, in the northeast also, you see a lot of green. These are the forests. So they are the malaria pockets. And these are the pockets which are very difficult to. The end game, we have lost once because we could not control. When we go to end game, when we try to eradicate malaria, we have to go to each pockets. So even one smoldering is just like a fire which can flare up anytime. So this is the history. The opening game, when we started malaria control, the greatest achievement was the discovery of insecticide. And if you see historically, The opening pre-partition, we had 100 million cases of India every year. That was before 1947. After 1947, we just from 100 million in India, we had 75 million. Where did the 25 million go? They went to Pakistan, because partition there was batwara of the burden also, as well as the assets also. So incidence in 1947 post independence was 75 million cases, and then DDT was invented by Paul Muller in 1939, and he got the Nobel Prize also. And once the DDT was invented, it was a remarkable invention, and it had a remarkable impact on the vector of malaria, the mosquitoes. By twice a year, thrice a year, spraying of the houses indoor with DDT, which was a residual poison as well as an immediate insecticide, it brought down from, say, you just see the from 1947, there were 75 million cases, and 1953 the National Malaria Control Plan was launched in 1953 based on the DDT spray, indoor spray. And in 1958, from 75 million, the cases came down to, say, around 2 million cases. So you can imagine the amount of reduction in India. I'm just talking of India, and the same thing happened all over the world. And the WHO and the world bodies, they got very optimistic, if we can reduce just by indoor insecticide spray by DDT. And Paul Muller got the Nobel Prize winning invention. From 75 million in India, it came down to 2 million. So they became very optimistic. In 1958, the program was changed to National Malaria Eradication Program. So they thought they will eradicate it. And the greatest success was in 1961. There were only 50,000 cases. And then it appeared as if eradication seemed to be within reach. Like an end game has come. Like in chess game, they say it is very difficult to win a winning game in end game. So they thought that it is. But however, after 61, uh, using the same strategies of indoor spraying, the cases kept on rising, and 1977, from 50,000 cases, again it built up to 6 million cases. And then they realized that we cannot eradicate malaria, and there were many reasons given. And then the eradication was abandoned in 1977, when there are 6 million cases from 50,000. And different reasons are given for losing the end game. That is complacency, that people got overconfident, administrative failure, the interiors were very difficult, transport was not available, insecticide. As I said, the tribal areas are hard to reach areas, and now there are a lot of disturbed areas, the tribal areas. So health services are not there, penetration is not there. Operational and technical problems were resistance of vectors to insecticides. If you overuse something, the resistance developed. And DDT, which won the Nobel Prize for the inventor, subsequently was found to have adverse environmental impact. It started accumulating in the food chain. DDT was found in breast milk, and lot of other concerns came up, and ultimately DDT was banned. So this that why this we lost the. That's why any new discovery, we think we are nearing the end game. So again, the current discoveries also we have to see in that context. Then resistance of vectors to insect, then resistance of parasites to chloroquine. This was a major issue, particularly falciparum malaria, which is the more dangerous type of malaria. Falciparum malaria, the chloroquine resistance was a major problem. That time in the armed forces, we started using quinine because we had a lot of failure with chloroquine, tetracycline, quinine combination, and subsequently now we have got the artemisinin combinations. So that has brought some improvement in treatment, and we could treat falciparum malaria. And then people started losing interest in malaria. Funds started reducing because they thought malaria is about to end. So many people switched over the carriers to other. So these are some of the reasons. So now we are again at the brink of a new game. The Bill and Melinda Gates Foundation put malaria eradication back on the agenda in 2007. And a lot of funding is being given. And earlier also, like impregnated insecticide nets, bed nets, impregnated with, I will come to that. We have done trials on that also in a tribal area. Global malaria eradication is complex and almost it is impossible. Thrust on technological solutions like insecticides, treated nets, indoor spraying, diagnostics, drugs, etc. These are good, but these are not adequate to eradicate malaria. We will see the reasons. Technological advances, vector control, and vaccines. Again, we are going. It is good to have technological advances, but they can be just one of the chess piece. You have to see the whole board, and like each game of chess is different. The pieces may be the same, but the game may be different. Similarly, what measures will work in India may not work in Africa. What measures will work in Africa or what measures work in Peninsular India may not work in the Northeast because of the different vector combination with different terrain combination. So, just like a game of chess, one has to take call at the regional epidemiological level. We cannot have one guideline for the world, one guideline for the country. We have to epidemiologically stratify the areas, and then these are technological advances. We will discuss about gene drive techniques, genetic modification of mosquitoes, spatial repellents, and the malaria vaccines RTSS, AS01, and R21. What is the status as on today? We will just touch on it. There are detailed papers on that, and how we have neglected the social and anthropological factors. And if we do it again, we may lose the end game. So I will just give a very broad overview of the present situation in India. We have made advances. In 2001, we were 2 million cases and about 1,000 plus deaths. Actually, these I feel are underestimates because most of the tribal areas, remote areas, they don't have even health services, disturbed areas, insurgency areas in the northeast, Naxalite areas in other tribal areas. So they are very poor data. So there are underestimates. Definitely, there may be more deaths and more cases because tribal areas go neglected, and here the fire keeps on smoldering of malaria. So then, migrant population from tribal areas, from these highly malarious area, people come for projects to urban areas. So they carry the gametocides. They are the mosquito parasite carries and may spill over to urban areas. 2015, it came down to 1 million cases, and 2022, just around 2 lakh cases, under 2 lakh cases. So it is a remarkable achievement, 2022, because perhaps of the social developments. And one of the important, I think, is like the Swachh Bharat mission and the toilet mission, that elimination of open air defecation, because I will tell some experience ki how it has been a neglected issue, that how malaria is related to open air defecation. So this is again the world map of malaria. Overall, what is the global situation? Malaria is basically associated with poverty and deprivation, poor housing, lack of toilets, and most of the reduction in our country is due to better housing, better toilets, cutting down on open air, reducing open air defecation, eliminating open air defecation. And as on today, globally, there are around 249 million cases around in 85 endemic countries and around more than 6 lakh deaths. 2022-23 latest figures, whatever is there. And 95% of the cases are in sub-Sahara Africa. So as on today, the major problem of malaria is Africa, and in India, it may be in the tribal areas. And most of the interventions and trials are being done on the vector control and vaccine in the sub-Saharan Africa, where it is endemic and highly endemic, moderate to hyper-endemic zones, and there it is required. So as I said, what is relevant there may not be relevant in our country where the transmission may be not that intense. So this tribal malaria I will be discussing particularly from the Indian context, residual transmission zones, regions for losing the end game, and the human dynamics as well as the vector dynamics are different in tribal areas. So this what we work say, we are talking of spatial repellents, insect repellents, or insecticide treated bed nets. So that is for mosquitoes which are resting indoors. As I said, there are different species of mosquitoes in India. There are 45 species of Anopheles mosquitoes in India. Out of them, about six are major vectors. And there are different urban area. There is a different vector, Anopheles stevensii. Rural area, Anopheles culicifacies. Then forest area, there are different vectors. Like in the teak forest, you have got Anopheles fluviatilis. Foothills, you have got Anopheles minimus. So if you have got urban vector, Stephensii, they rest indoors. So you can use the residual spray and the spatial repellents or insecticide treated bed nets where you can prevent yourself from getting mosquito bites. But the problem is in the tribal areas where most of these vectors, Anopheles fluviatilis, they rest in the trees, they rest outdoors. So they are not amenable to spray because they are out of reach of spray, and in the open, the spatial repellents will get dispersed. It will not have an impact. So therefore, insecticide treated bed nets do not work. I have been posted in a tribal area. We carried out a trial that time in, I think, 1999, where newly the insecticide treated bed nets had come. And we found no impact. Some villages we gave the bed nets and some did not. So there was, because the vectors were resting outdoors, because the tribal area, the vector was Anopheles fluviatilis. So those and people who are getting malaria are people who are working outdoors in the night due to occupation or due to these tribals. They go to the forest at night for many types of activities. Sometimes they are distilling alcohol. Sometimes they are smuggling teak wood, the teak forest. So a lot of illegal activities in the night time, and they were exposed to the fluviatilis, and they were bringing back the yeah, and there were tribal villages also and tribal forests also, and they were bringing back to the villages, and in the villages we found a different vector, Anopheles culicifacies, and the local transmission near the habitation was done by the culicifacies. So there was a international flight was from the forest, was from the fluviatilis, and there was a domestic flight also by the culicifacies, the connecting flight. So these are, we have to study. The field work is lacking nowadays. Many of the time models are made from the computers. I call them laptop epidemiologists. Field epidemiology is missing. When you do the field surveys, you find different type of. Then we found different type of human behaviors also. I'll be discussing that very briefly, just to give an idea how complex it can be. So dynamics in tribal malaria, understanding the time in tribal transmission is entirely different than urban transmission. Tribal population in the forest holds center stage for endgame for various social and anthropological reasons. So, in tribal areas, the falciparum rates are always high. Vectors such as fluviatilis and dirus. I said fluviatilis in central India teak forest, and Anopheles dirus is in the northeast forest, and that Anopheles dirus also rests outdoor. Earlier it was called Anopheles balabacensis. Now it is called Anopheles dirus. They also behave like Anopheles fluviatilis, outdoor resting, and then both this Anopheles fluviatilis and Anopheles dirus both are very prefer the human blood, because some anophelines are not very efficient vectors because they prefer to bite animals like culicifacies. In rural areas, they are not very efficient because if the cattle is there, they prefer to bite cattle. But their very high density of culicifacies, they will bite humans also. Or if the cat animal is not there, so they prefer animal blood. But whereas fluviatilis and minimus, even with very low density, they can transmit very efficiently because they prefer the human blood. So these are the fluviatilis and Anopheles dirus are the vectors in tribal areas. Marginalized due to various is inaccessibility, and these tribal areas are inaccessible to healthcare. They do not get treatment, and this tribal population, they have a very high infant mortality rate because children, they die of malaria, and those who survive, they have developed some sort of preimmunity. Pre-immunity. It is not a very robust immunity. It is by repeated in parasitemia. There, those who survive, they have some sort of. So then, adults, they do not suffer. There are no outbreaks of malaria because there is a sort of some stability, and which is not a very strong immunity. Kacha immunity. So that's why I am not very optimistic of vaccines, because nature has not given immunity after repeated infections. It solid immunity. It gives some immunity, that is immunity, which is not very long lasting. So vaccine also tries to imitate nature by injecting the same antigen and the antibody response or the immune response or T cell, whatever. So once the natural infection is not long lasting and robust, so vaccine we should not expect it to be a game changer. It can only be a crutch to tide over the crisis, and particularly these vaccines are recommended in Africa in the children, because as I said, the children are vulnerable, they die in the infancy. So if that period is covered with the vaccines, it tides over, they become. Yeah. But as this immunity, we don't know how long it will last, vaccine immunity, because the natural immunity, those tribals who come out of those areas after some time, if they are not repeated challenges, they lose the immunity. So they become equally vulnerable. And in these tribal areas, there are the local population develop immunity, and some troops come. They are naive, they are not having immunity or pre-immunity, we call it. So outbreaks occur in this newly inducted people into endemic areas. Local people do not suffer, but there is unstable malaria, large outbreaks occur among the troops or any other migrant population who are coming. Or similarly, if a group of tribals go into a area migration, lot of vulnerable people, it may, if the vector is there, conditions are there, it may trigger an outbreak. So these are some of the subtle things which we have to observe. And from natural history of disease, when we find the immunity from natural infection is not. So these are the problems, whether it is tuberculosis or whether it is malaria, where mostly cell-mediated immunity, which is very unpredictable. So vaccines are a temporary crutch only. Long term, we have to improve the conditions, overall ecological conditions, social conditions. Malaria is more a social disease, and one has to combine the social issues with the technological advances. Then lifestyle and socio-anthropological reasons and beliefs, like treatment seeking behavior and other things. So as I said, the toilets we have seen in this uh, tribal areas, and we found troops also when they move in camps in these jungles and forest, they also have makeshift toilets in the near the forest, near the streams. So they get exposed to mosquito bites when they go for call of nature. And this have been reported from some other countries also. So those earlier days, the majority of the rural and tribal people lacked toilets, and they used to go for open air. And nomadic and forest related activities. So these, as I said, malaria has come down, like we have cut down, we have tried to eliminate open air defecation. So that is a major thing. So that I find very little mention of that when we talk of malaria, but that is important. So these are some of the studies in African countries, in Ethiopia, Cambodia. They found that if the areas, poor areas, poverty, forest, they go for call of nature, and any toilets or other sanitation in the villages and none in the forest farms. Many people depend on the forest for their occupation, and there have been reports by qualitative research. These people report that they are very bothered by mosquito bites going to the toilets and several parts of the body, and particularly when they expose their lower part of the body, which has very thin skin, and their mosquitoes very easily can bite and transmit. And and most of the people, they go during the peak biting time, late night or early morning, and most of the anopheles mosquitoes, they bite during dusk or dawn, late night. So there is a lot of synchronizing of the mosquito habits and the human habits in the forest. And as I said, these tribal areas, these vectors of mosquito, anopheline mosquitoes, they rest in the outdoor. So if you may spray the houses, but the transmission is taking outdoors. So uh, controlling malaria just by technological measures such as indoor spray and insecticide treated nets might not achieve elimination. Well, those when the insecticide treated nets are mosquito nets which are impregnated with usually pyrethrum derivatives, insecticides which are derived from pyrethrum. So what happens ki earlier people used to sleep in mosquito nets, and when they used to get up in the night the mosquitoes used to enter the net and bite them, or they are, but if it is treated with insecticides, they repel. It acts so even if the it is pulled up, the mosquito does not enter the net because it is repelled by the insecticide, or when it comes in contact, it may kill as a residual poison. So that was the very successful where in urban areas there is Anopheles stevensii or even Anopheles crucifix, they are resting indoors. But as I said, they have minimum impact on the forest malaria because the vectors are resting outdoors. And as I said, there are many vectors. I mentioned few only. Crucifix is rural areas, urban areas, stevensii, northeast forest, Anopheles dirus, Anopheles fluviatilis in forest of central India, Anopheles minimus in the foothills. Then coastal areas, they have got Anopheles sandaicus, which can withstand some salty water also. So there are different areas, different regions, and each have got their own uh, resting habits, breeding habits, biting habits. And based on these things, there is a need to reimagining malaria to incorporate the association of human and vector behavior when going for malaria elimination. You see, we have got the vector control, new vector control measures are emerging, like gene drive or the spatial repellents, and many. It has come in the past also. It's nothing new. So it until unless we combine everything, we use it judiciously, depending on the dynamics of the vector, we are not going to achieve elimination. And there are different studies in Ethiopia that people without toilet facilities were more at risk for malaria transmission of controlling for other factors. Similar findings from Nigeria. So these are, as I said, Africa is the major belt of malaria. So we have to see vaccines is supportive, one aspect in the toolkit, but overall conditions of living, sanitation, toilets will bring down malaria in addition to this vector control and vaccines. Then troops, as I said, similar what the nomads, what the tribals, so most of the troops, those who are working, those who are in operations rather in the northeast, they have to camp in the jungle. Sometimes they have to camp, they have to march there. And some of the stations are surrounded by tribal belts. And transmission, very intense transmission. And as I said, the troops are not natives. So they do not have immunity which the natives have. So they become highly vulnerable to malaria. And I had been there in a tribal belt. We carried out a insect impregnated mosquito nets. And as I said, we did not find any impact. Then later, we did the vector studies. We found the main vector was the fluviatilis and the main people among troops also. Because we had a military station, and people who are staying deep in the military station, the families and other soldiers, they were not getting affected by malaria. Then there was some military platoon which was in the periphery, and they were doing patrolling in the forest, and only they were getting malaria. Very high level deaths also, very high level, because as I said, they were in contact with the tribal villages and the forest, and the vectors which were outdoor, and they were going now night duties. So they were highly vulnerable, and they did not have the immunity. So we had to do a visit their barracks, talk to them, and we studied their habits also, and we found they were highly vulnerable because there also they had some behavior problems which made them vulnerable. So this is the central India. We did a study when during my army career, and they were in the Satpura ranges, and it had an altitude of plus 700 meters, and it was surrounded by tribal mostly and non-tribal villages. So the peculiar problem was the perennial transmission of falciparum malaria. And DSC personnel was the people who were doing the guard duties, Defense Service Corps. And among the civilian, we found the tribal villages. There were some villages were tribal. They were most affected with falciparum malaria. They were going to the forest and bringing back the malaria to the other villages. So this is the station roughly I have seen, and these are the surrounding villages. And the soldier who was doing guard duties was, these are the different villages, and these were the teak forests. There were streams flowing in the forest, and at the edges the fluviatilis breeds. So we identified the larva, we collected the larva, the eggs in the, we grew them, and we identified the adult as fluviatilis. So these, and in the villages there were the pools of water, and from here we identified larva, and subsequently when it matured we found it was culicifacius. So there were two vectors, and when there are two vectors the transmission becomes very intense. So one vector was bringing the malaria from the forest to the village, and from the village the villages were affected by culicifacius. It was the main vector, but the main fatality was in the forest because of the falciparum, particularly the guards. Natives, they got adjusted. So this was the actually the layout. This outermost circle is the teak forest, fluviatilis, and the innermost green area, the tribal villages, the culicifacius, and the innermost is the blue area, the Air Force station. And most of the people staying in these Air Force stations, because we were using impregnated mosquito nets in the Air Force station, all were using anti-mosquito measures, bed nets and all. So there was hardly any malaria because the culicifacius, as I said they rest indoors. So if they rest indoors, we were spraying the houses, and also we were controlling the people staying in the culicifacius belt. But those who are the soldiers doing the night duties, patrolling in the forest, and the tribal who were going there, they were exposed to Anopheles fluviatilis, which were resting outdoors. So these are some of the measures. These vector control measures are nothing new. There have been many vector control measures in the past which have been as effective as what are the newer methods coming. But anything in isolation doesn't work in malaria. Everything has to be combined. Depending on the biodynamics of the vector and human behavior, deweeding of water channels, because wherever there are weeds, there is some stagnation, and where the breeding of mosquito takes place, particularly fluviatilis. Delta method impregnated nets. It had no effect outside the tribal. Inside the Air Force station, it had effect. But when we did it in the tribal villages, because most of the tribal villages were getting infected outdoor. Indoor residual spray also not much effect in the tribal villages, but it had effect inside the Air Force station. Then larvivorous fish. There are certain fishes, guppy and gambusia fishes, which eat the larva of the mosquitoes. So mosquito larva is also a good target to attack because they are accessible. Whereas the adults rest in the air, if you can attack the larva, you can bring down the density. Then the insect repellents. I will explain the bottoms up approach. As I said, when we identified that the soldiers who are going in the forest in the night, they are the people, we could make out that they were getting exposed due to bites by Anopheles fluviatilis. They were resting in the treetops canopy. And then we advised them to use the Odomos or other repellents on their exposed parts and hands. And day and night we were telling them every 4 hours to put when they go. But still it did not bring it down. Then once we had gone to inspect their living conditions, and most of the barracks we found that the toilets were not having adequate water, so it was not being easy to flush. And we asked them, what is the problem? He said, no, the water is less, so we can't use the toilets. So we asked them, then what you do? So most of them, I'm talking some years back, they said, we just go to the forest to relieve ourselves. So that gave us an idea that perhaps we are doing everything inside the barracks and all, but since they are also not having the good toilet facilities, they are also going to the forest. Most of them come from the village background, and those days the open air defecation was a routine for them, and they were going near the streams where the mosquitoes used to rest and breed, and they were exposing their bottoms early morning, late evening. So that is the time they were getting the bites. So then we advised them, okay, we will try to repair the toilets. In the interim, you use one Odomos for the face and one for the bottom. We gave them an extra Odomos, ki at least when you go to really use it on your bottom. And that really brought down, besides the other measures. So one has to go in depth to find the problem, where the problem is. And then, of course, because most of them may not, change of behavior is not easy. We have seen in some tribal areas, the toilets are there, people still go outside because they feel more comfortable. So it takes time for change. So these are some of the results we got. The This is the rate, villages and air force station is the, blue color is the troops. So first and second year we find that the blue color bar is taller than the villages, that is the natives. As I said, the immunity is not there. And once we identified the problem, we gave the Odomos and other things for the bottoms. And so if you see the third year, that is the third year is when we could identify the problem and implement the malaria control measures. The villages, the malaria control is increasing, brown bar, but we could check it in the Air Force station. Similarly, here is the brown tall bars are the people who are doing guard duties, Defense Service Corps. So compared to other Air Force station, they were having the highest rate. But by the third year, their rate also came down. Of course, not compared to the other Air Force station people, but still we could, to great extent, we could check it. And these are the tribal villages, that these are the tribal and non-tribal villages. The brown are the tribal villages, and the uh, black gray is the non-tribal villages. So tribal people were going in the forest and they were getting the malaria, and intense transmission was taking place, and it was difficult to control. So again, this is the tribal and all. So these are, as I said, the epidemiology is actually field work. But why it is field work? Because of the diversity of the ecology in different parts of the same country. As India is a big country, Africa must be much bigger. And sitting in India, I cannot say what is happening in Africa. It may be a different board game there, because the vectors are different in Africa. In India, I said we have got 6 vectors, main vectors: Culicifaces, Stephensii, Fluviatilis, Minimus, Sundicus, Balabacensis, Dirus. But in Africa, they are different. Anopheline. They are anopheline, but they are different species. So their behavior may be different. You have to study their people behavior, you have to study their, and then plan the anti-malaria measures. In isolation, nothing works. So that is the actually Sioux leather epidemiology. By visiting, that was known as Sioux leather epidemiology, by visiting the people, talking to them. Now what is happening, the solutions are made in the lab, or the models are made in the computer. I call them laptop epidemiologists, instead of Sioux leather epidemiology, and it is projected as if this will work everywhere. It doesn't work like that. As I said, malaria is like a game of chess. The pieces may be same, but each game is different. So African game may be different, Asian, Indian game may be different. So what I have said is from first-hand experience from a tribal area. It again may be very limited experience. In some other area, I have to think of other strategies. So this, from the Sioux leather epidemiology, we had the armchair epidemiologist. Now we have got the laptop epidemiologist. So these are, and if you see, the government has said that open air defecation is has been eliminated in Chhattisgarh. This is from, but these are the pictures which are showing that tribals, some of them still prefer to go in the open air because the behavior change takes time. So these are again, this is a common, yeah, early morning. If you are going by train now, of course it has come down, but occasionally you will see there is a lot of urban migration. The sanitary facilities are not adequate, so the exposed bottoms are the good site for mosquito bites. So these are some of the gaps one has to plug. And now we come to whatever the recent advances are there in malaria control. One is the lot of talk is about the genetic modification, gene drive technology. This is actually some genetic modification in mosquito populations to restrict the ability to transmission by using CRISPR technology for gene editing. This is becoming very popular, and they say that anybody with access to this can do the editing. So, but it may be just like a toy in the hands of a child. So it has got a experiment superiority over the other vector control measures like insecticide treated bed nets and residual spray. And how the intervention works, there are different methods. One of the methods is that uh, it strategy being evaluated by some gene editing. It changes the proportion of the mosquito population, and it increases the proportion of males compared to female mosquito, because the female mosquito transmit the malaria. So it, by some gene editing, it increases the male species of mosquitoes compared to the female. It causes a adverse sex ratio. It's unfortunate, one would say that up till now we were struggling with human sex ratio, which was not favorable to. But now they are trying to do gene editing to do the sex ratio imbalance in mosquitoes also. So this is one, and other also other pathways are there by gene drive, gene editing, by which the transmission capacity of the parasite in mosquitoes. But these are, there many are slip between the lab and the field. You see, idea is to make this gene editing and release these mosquitoes so that they, but as I said, it's very vast. I mean, indoor and other mosquitoes. But when you see the tribal areas, most of the mosquitoes, vast forests, they may be very high preponderance. It may not reach. You see, even 30 years back also we studied sterile male technique, that they used to sterilize by radiation males and release them. It has not made any game changer. It never proved to be a game changer. So these are very good lab experiments. So it is very difficult to predict which terrain it will work and how much it will work in actual practice. So, and there are other uncertainties over sustainability and impact on non-target species, because you are interfering with gene. There are ethical and regulatory concerns over biodiversity of species, because in nature everything is balanced. So if you interfere, they may be, we have to closely watch. There may be regulatory and ethical issues. This is a paper, I will be giving the reference. There is a whole paper, recent paper on this gene drive. In the end, I'll be giving the reference. So those who are interested in go in that. I am not going into great detail. Then there are other repellents which have been coming again and again. And insecticide treated nets was a big thing. It was thought that it will change, but it had no impact on outdoor resting species. Then spatial repellents like mosquito shield containing transfluthrin. It is on the principle that usually bed nets you use at night. Then they observed that many mosquitoes have changed their biting habits. When you are sitting in the evening also, they will bite. So there are certain spatial repellents which will, if they are sprayed, there is some residual effect in the space. It will have some translucent base. So again, this same thing applies for outdoor vectors. It may not. And main problem of residual malaria is the outdoor malaria and tribal malaria. Then these are the malaria vaccines. This is again, and again I would tell you, when I was doing my post graduation in 1985, we used to have seminars on malaria vaccines. Since 1985, we are thinking that vaccine is around the corner. That is 1985, 20, almost 50 years. We are still waiting for a malaria vaccine. Yes, they have developed it now, but we don't know. As I said, nature has not conferred immunity after natural infection, which is long lasting and robust. So whether we can replicate it is questionable. So whatever it is, the position as on today is there are two malaria vaccines which has been approved by the WHO to be used with discretion in endemic areas and in children, 6 months to I think 37 months. And because children are most vulnerable in hyper-endemic area, those who survive, they develop some immunity. So RTSS AS01 malaria vaccine in infants and children targeting circumsporozoid protein. So it is attacking the pre-erythrocytic phase before it enters the red blood cells. You see, malaria, you must have, the cycle that mosquito bites, inject the sporozoids, then goes to the liver, and then seasoned and erythrocytes are infected, and that cycle. And so it before it enters the erythrocyte, it is targeting the vaccine. Phase 3 RCT in sub-Sahara Africa. That is one vaccine. Other vaccine is R21 matrix M in African children, circumsporozoid protein fused to hepatitis B surface antigen. This is also phase 3 trial. And the WHO position is that the use of malaria vaccine for prevention of P. falciparum malaria in children living in malaria endemic areas, prioritizing areas of moderate and high transmission. So it is just to tide over the crisis, and the efficacy of 3 doses is, I think, less than 50% or 50%. And the fourth dose is recommended. Efficacy comes up to 70-75% in the phase 3 trials. So this has to be seen how long it will. Definitely doesn't long lost because number of doses are more, 4 doses, and then every year booster perhaps. So it's a temporary measure. And efficacy, like both malaria vaccines have been pre-qualified WHO in phase 3 clinical trials. Both vaccines reduce malaria cases by more than half. First year period more at risk. Fourth vaccine in the second year of life, prolonged protection. Both vaccines reduce malaria by 75%. That is after when given seasonally in highly seasonal transmission where seasonal malaria chemo prevention is provided. Chemo prevention, there are some places you are giving chemo prophylaxis with drugs like Daraprem or chloroquine or doxycycline. These are the drugs for chemoprophylaxis for visitors. So it has a temporary effect. It won't be very long lasting, and side effects are there like convulsions, fever, but they say it is manageable. So again, we have to wait and watch. So conclusions is malaria is a complex disease because of human behavior, vector behavior, parasite resistance, environmental conditions, different permutation and combinations. So you have to study the dynamics at the local level. Need to revert back to shoe leather epidemiology. You have to get out in the tropics, uncomfortable weather, uncomfortable terrain, hostile people. These are also the challenges. Supplement quantitative studies with in-depth qualitative studies of human behavior and related ecosystem, collaboration with social scientists and local population. This is the biggest gap which we have had in the past and we may have in the future if we do not take this into account. So these are the references as reimagining malaria, IL. You can see at your leisure, Nigeria. Then these are some of the references. Then, as I said, reference number 7, we did the delta metrin impregnated bed nets study. We found no impact because we found the vector was outdoor. Then entomological correlation we did, tribal malaria. So we brought out number of papers on malaria from tribal areas, which were quite informative and educative to us also. And now if you see the last number 11 is the reference on gene drive technology. So those who are interested on the newer technology can see reference number 11. It is in the public domain, open access. Then 12 is the paper on insecticide repellent, spatial repellent. Then 13 and 14, the last are the malaria vaccine. One on RTS vaccine, other is R21. So these are the references. I have finished. Thank you. I think I can stop sharing the screen.
It was an absolute pleasure listening to you. Very well presented session. Lot of takeaways for the audience. We have a few questions from the audience. With your permission, let's go on to the first question. My first question to you is, are there any particular population, example migrant workers, tribal population, children, that you feel remain vulnerable or underserved in malaria prevention efforts?
Yeah, definitely, particularly the tribal hill area, because the health services are limited there. Sometimes microscopy may be limited, drugs may be limited. In fact, when I have served in a tribal area long back, that time they run out of anti-malarials, and we had to give them from the military hospital. We have to give them. So there are issues like penetration of health services. Most of the health services we are having in urban areas, and we are neglecting the remote areas. And we cannot eliminate any disease if we neglect those areas, because they will always be the seeding for the flaring up, and which has happened in the past also. As I said, 50,000 cases it came down and then suddenly started rising. So definitely tribal areas are actually the center where we have to, and these populations keep moving all over the country also. So they keep transmitting malaria to other parts.
Well explained. Moving on to the next question. So my next question to you is, how do you approach a differential diagnosis when malaria mimics viral fever or dengue, especially in endemic regions?
In endemic regions, we had a protocol. We will do first any fever case. We have to exclude malaria. Had a microscopy those days. Even now, that is the gold standard. And then we have got the rapid diagnostic test. And another thing is there, particularly in falciparum malaria, it can be very deceptive clinically also, because the parasites goes down into the deep vessels, in the cerebral vessels or here, and in the peripheral vessels it may keep on coming negative. There may be hardly any parasites in the, and we may get a false result, and the parasites may lodge in the deeper vessels. So our protocol was ki any person with high fever and comes into, say, giddiness or dizziness or even in coma, we gave anti-malarials irrespective of the, particularly to serious patients. The patient is moving around, okay, we can wait and watch. But all serious patients we treated for malaria also, for cerebral malaria. Like now, of course, those days we used to give quinine and the tetracycline for resistant malaria, falciparum malaria. Nowadays we have got artemisinin combination. So the way to save the patient is to give anti-malarial in endemic areas. Person comes with fever or serious.
Thank you for answering it elaborately. We'll move on to the next question now. So my next question to you is, what can private practitioners and general physicians do to contribute to malaria elimination in their communities?
Definitely, they can do a lot because they are in touch with the community. Firstly, they can educate the people about the prevention of mosquito bites, and not only prevention, how to control the breeding of mosquitoes. Wherever there is water collection in urban areas, sometimes they are open overhead tanks where the covers are missing. So these Anopheles stevensii, they breed in this type of clean water, overhead tanks, or there is water collection. So they can educate the people, the other authorities, public engineering services, to have good water management so that the mosquito density is less. Then advocate mosquito nets, and then of course they any case of fever, they should do a test for malaria, and if they find positive, they should notify malaria cases to keep a track of and report to the local health authority, so that any early outbreak or anything can be investigated and controlled.
Thank you so much for answering that. This brings us to the end of this session. Thank you so much, Dr. Amita, for your time and effort in putting together this interesting session. I hope our audience enjoyed your session thoroughly. To our dear viewers, thank you so much for attending this session. Do share any more questions if you have. Until we see you again, take care and happy dog plexing. Thank you.